My name is Oktay. Today's video is about peptides and proteins. There are three big groups of compounds in nature. You've got carbohydrates, fats, and proteins. The building blocks of proteins are the amino acids. Here are three examples. The simplest molecule is glycine. You got a C atom with four groups. All amino acids have a common structure. One group is amino group, another group is a carboxyl group, third group is hydrogen, and the fourth group is different in each amino acid. In the case of glycine, it's just another hydrogen atom. There's only one type of glycine molecule. With the next bigger molecule, there's a difference. Because now you got four different groups at this central carbon atom, and this means you have this phenomenon called chirality. There are two sorts of this molecule. They are mirror images of each other, like the right hand and the left hand. This left compound is called L-alanine, and it's a mirror image called D-alanine. The peptides and proteins are made of L amino acids. The D amino acids are also important because they are the building blocks of bacteria. This is why yogurt is very healthy, because it contains several percent D amino acids. The human body contains 37 trillion cells. This number is exceeded by the number of microbial cells, which is 39 trillion. These are bacteria, viruses, and fungi. Because these microbial cells are so tiny, they make just 1 to 3% of the body mass. There are several ways to group amino acids. One is in essential and non-essential amino acids. Essential means that you have to provide these L amino acids because the body can't make them for itself. The non-essential amino acids can be made by the body from other amino acids. That's the difference. And you can see another way to group these amino acids is by their side chains. Some amino acids have charged side chains, positive or negative. Some have polar but uncharged side chains. Some are special cases with special atoms like sulfur or selene. And others have hydrophobic, this means non-polar side chains. This is how peptides are made. Each amino acid has at least two functional groups, that's amino group and the carboxyl group. Amino group of one amino acid can react with the carboxyl group of the second amino acid. The free electron pair at the nitrogen connects to the carbon atom. One molecule water is separated as byproduct, and you get this group. This is called amide group, and this molecule is a dipeptide because it consists of two amino acids. And this amide group has here at the nitrogen a free electron pair, which is part of a pi electron system, conjugated pi electron system. And this group is stabilized by mesomeric effect. And this dipeptide has again two functional groups left. So a third amino acid with its amino group could connect here, then you get a tripeptide, and so on. This is the code of life. That's a DNA molecule, deoxyribonucleic acid. It consists of 4 letters, of 2 complementary pairs of bases. Guanine and cytosine are a pair, and adenine and thymine are another pair. And this is the map of all 21 amino acids and the code of these amino acids. For example, L-leucine is here. You have to start in the middle. So A, AA, or AAG, that's leucine. Alanine is here. Start in the middle, GCA or GCC. All these are the codes for L-alanine. And you can see there's also a code for stop. When the protein is finished, then this production stops, and you get the protein molecule. This is an example for peptides. Peptides consist typically of 2 to about 50 amino acids. That's the difference between peptides and proteins. Proteins have over 50 amino acids. And this is a simplified ball and stick model of the peptide glucagon. Contains 29 amino acids, and it's a hormone. Hormones are messenger molecules, and glucagon makes a pair with insulin, which has 51 amino acids. And you can see the blue atoms are nitrogen. That's the amide group. Here's another one. And the pair of glucagon and insulin are a feedback system for the glucose concentration in blood. Whenever the glucose concentration gets too high, then the messenger molecule insulin makes sure that the concentration goes down. And when it's too low, then the hormone glucagon kicks in and starts the production of glucose. And this system makes sure that the concentration of glucose in blood is in a certain concentration range, which is healthy for the organism. One example for proteins are enzymes. On the top you can see how an enzyme works. It is a biological catalyst. The substrate connects to the enzyme. It connects to the active site of the enzyme, this part. When the enzyme binds the substrate, it loosens the existing bonds and makes the reaction happen. And finally, the enzyme releases the products. And now it's ready for the next substrate molecule. At the bottom you can see how our inhibitor works. Inhibitor blocks the active site of the enzyme, and now it can't work anymore because it can't bind the substrate. Viruses have enzymes too, like for example the HIV virus has a protease enzyme. And this strategy, inhibitor strategy, was used successfully against the HIV virus. The inhibitor blocked the enzyme of the virus. Now it couldn't replicate anymore, and this means the virus is no longer able to weaken the immune system. That was a successful strategy. It did not stop the infection, but it stopped the weakening of the immune system by the virus. These are the different structures of big protein molecules. You have the primary structure, which is a linear sequence of amino acids. Then you have the secondary structure. This is a 3D form of local protein segments. The tertiary structure is the 3D shape of the protein. And the quaternary structure is a big structure of folded proteins in a multi-subunit complex. These are the bond types that make the tertiary structure of a protein. You have the hydrophobic interactions. These are van der Waals bonds. They are weak. And you have the disulfide bridge. This is much stronger because it's an atomic bond. Then you get hydrogen bonds. Then hydrophilic interactions. These are hydroxyl groups that are oriented outward to be able to make intermolecular hydrogen bonds with water. And you got ionic bond. This is how the protein biosynthesis works. It starts at the nucleus of the cell. That's the first step, is the transcription process. It makes a RNA copy of the DNA segment. This RNA copy is modified, exported out of the nucleus. The ribosome takes the messenger RNA and transfer RNA. For the synthesis of the proteins, and afterwards there are modifications. The protein is folded, and then it's ready. So the ribosome is the protein synthesis machine. This simulation is fantastic. Shows how this works. In green and yellow you can see that's the ribosome. This is the mRNA, messenger RNA. In dark blue you can see the transfer RNA. The transfer RNA brings one amino acid after the other to this production process. And you can see that, there are other proteins involved, and these are the light blue proteins. Protein folding happens in the endoplasmatic reticulum, which is a network of membrane-limited channels. This will you see soon in this animation. Here's the protein that's synthesized, and eventually it will come out. This is the product of 3.8 billion years of evolution that's made this machinery. This is an example for a big protein molecule, that's keratin. You can see it has a superhelix structure. And one thing about keratin is it contains 14% cysteine. Cysteine is the amino acid with the SH group, sulfur group. And these sulfur groups make the disulfide bonds. And keratin is the protein that makes the stable hair. And you can change the structure of the hair by making permanent waves. The first step here is open this SS bonds with a reducing agent like ammonium thioglycolate. Then you get two SH groups. Then you put the hair into the new shape, form it into the new shape. And the last step is to treat the hair with a neutralizer like hydrogen peroxide that restores the disulfide. Then you have the permanent wave. This is something very important in the world of proteins. That's the protein domains. You can see big protein molecule with different areas and different colors. These are the protein domains. They are self-stabilizing parts of a protein, typically contain 50 to 250 amino acids. They're structurally independent from the rest, and each protein domain has its own function. This is an example of the protein domain. The coronavirus has a protein with a protein domain. That's the spike glycoprotein, this here symbol S. Its function is to enable the virus to enter the host cell. The coronavirus has several other proteins. The M and E protein, they are important for assembly of the new viruses. And the N protein is a soluble protein. It is a package of the RNA genome. This is another example of proteins. It's hemoglobin. It's the transport system for the oxygen in the organism. It has two components. The protein component is the globin here, and in the animation you can see it has a flat structure. That's the porphyrin ring. That's the second component. In the center of the porphyrin ring there's the iron 2 ion, and this iron 2 ion binds the oxygen molecule for transport in the blood. This is the biggest protein in the human body. It's Titin. Its function is the muscle contraction. Consists of 34,350 amino acids. It has 244 protein domains, and it makes about ½ kilogram of the human body. It's a molecular spring, and it stabilizes the muscle. And you can see it's nicely embedded in this filaments. This was a great success of AlphaFold artificial intelligence. For all these applications and for new drugs, you need an understanding of the 3D structure of these protein molecules. And this picture shows in green experimentally found structure of these molecules, and in blue the structure predicted by the artificial intelligence AlphaFold. And you can see this really nicely matches, except for a few exceptions. And every single bond can rotate. This means if you have a very big protein molecule, you have a very high number of possible conformations. This number can reach 10 to the 300. And for comparison, the number of the atoms in the universe is estimated to be between 10 to the 78 to 10 to the 82. But still, the AI AlphaFold was able to determine the structure of all these proteins, and this is a great scientific breakthrough because it will lead to potentially new drugs. And this AlphaFold is already used for proteins of the COVID-19 virus. And that was today's video about peptides and proteins. Thank you for watching. Have a nice day. Bye-bye.